Just to introduce Dr. Joe Anderson, another new member of our team, which is very exciting. So we have lots of new people that we're introducing to you today. Dr. Joe Anderson also started working with the foundation approximately 2 years ago through the generous donations of the Manier Lapastel Foundation. And he presented to us some very amazing data looking in other disorders, which I'm sure he's going to share with you today, as a new approach for gene replacement therapy for other neurologic disorders and what that meant and what was possible for Angelman syndrome. And so Dr. Anderson is an associate professor at the University of California at Davis Stem Cell Program. He currently develops cell and gene therapies for various diseases, including Angelman and other lysosomal storage diseases and HIV. He's trained in preclinical and regulatory procedures and allows his team to move these types of therapies forward to human clinical trials. So we're very excited to have him here, and we welcome you with open arms.
Thank you, Allison. Thank you all for being here, and thank you to FAST for inviting me here to speak and present this new strategy that we're developing as a treatment for Angelman syndrome. So the idea is called cross-correction. So prior to my lab working on Angelman syndrome, we are developing stem cell gene therapies for lysosomal storage diseases, including Tay-Sachs and MPS I. And one day in the lab, the stem cell program is a huge lab with a lot of teams and individuals that work on different diseases. And one day, a grad student in Dave Siegel's lab and I were talking, and he was explaining to me his project on Angelman syndrome. And I had no idea about Angelman syndrome or UBE3A, and so we were talking about it. He was saying, you know, it's a one-gene disorder, UBE3A is deleted. And since these other lysosomal storage diseases that I work on are also monogenetic diseases, I thought, well, why not try this cross-correction strategy for Angelman syndrome? Even though it's not a lysosomal storage disease or a leukodystrophy where this strategy is being developed and used in the clinic right now, but why not try it? And so the idea of cross-correction is that you have a genetically modified cell. So it's the cell on the left, and it overexpresses the wild-type form of this protein that the other cells are missing. And what this cell will actually do is transfer that functional protein or enzyme to the affected cells, and thus restoring that function that's deficient in those cells, neurons for Angelman syndrome. And so that's what we proposed in our grant application. Dr. Siegel, Dr. Silverman, and I as collaborators, and we're grateful that FAST funded it. And so we then had to think about the design of how we were going to, I guess, make this strategy possibly work for Angelman syndrome. UBE3A is not normally secreted from cells like these other proteins are for these other lysosomal storage diseases and leukodystrophies. So we had to sort of manipulate UBE3A and modify it in a way that it would be able to be transported from these genetically modified cells to the affected neurons, because normally UBE3A is intracellular. So we had to modify UBE3A to allow it to be secreted from these cells, but then to also be absorbed into the neurons that were nearby. So this was something that we initially had to work on and test, and I'll show some of that data in some other slides later. And so the type of gene therapy that we're using is an insertional gene therapy. So this is where we would actually insert the functional copy of UBE3A into our donor cells, cells that were on the left of that one slide. So you have a cell with no UBE3A. We then take a functional UBE3A gene and we insert it into that cell using insertional gene therapy. and now you have a cell with functional UBE3A. The way that we do this is with a lentiviral vector gene transfer system. So this lentiviral vector is a normal human virus that will infect human cells, but it's modified so that all the disease-causing genes are deleted. So its function is to deliver a gene that you want into the cells of interest, and now you have this functional gene inside of the cells. And the great thing about lentiviral vectors is that there's permanent integration. So the way that the virus works that they've manipulated to make this lentiviral vector is that it integrates into the DNA, and so the gene that you put into those cells is permanently in there. So in theory, if you get enough cells gene modified, it could potentially be a one-time treatment because those cells contain that functional copy of the gene. And so the way that the cross-correction strategy works is that my lab and other labs that work on these other lysosomal storage diseases and leukodystrophies that are actually in clinical trials now, they use the blood system. So the blood goes all throughout the body, and so you're able to, I guess, deliver that functional copy and that functional protein everywhere in the body. So that's a great advantage because there's not only neurons in the brain, but you also have all your peripheral neurons. So being able to provide that functional enzyme all throughout the body is definitely an advantage of using the blood system for that cross-correction strategy. And then we had to think, well, what cell should we genetically modify? Like use that lentiviral vector to permanently integrate into the DNA of our cell to then deliver that functional enzyme to the affected cells. And so this is a classical chart of blood formation. So at the very top, you have this hematopoietic stem cell, so a blood-forming stem cell. We all have these in our bone marrow. A small percent circulates in our blood. And these cells know to home back to the bone marrow. So it's typically like a bone marrow transplant. So what we can do is specifically target these blood-forming stem cells with this lentiviral vector that carries the functional copy of UBE3A, and then transplant them back into the patient, and then every cell downstream, so all the cells below that top cell, can be made. So we continuously make new blood cells for the rest of our lives. Like a red blood cell has about 120-day half-life. So you have to continuously make new blood cells, new immune system cells for the rest of your life. So a great cell to target for gene therapy is the hematopoietic stem cell or the blood-forming stem cell. So the way this would work is that we would isolate the stem cells from a patient. This is routinely done. Bone marrow transplants have been done, I think, since the 1950s. So it's routinely done for cancer patients and now for a lot of gene therapy. There's already been trials for beta thalassemia and ADA SCID where they've actually treated infants. So Don Cohn's lab down at UCLA, they've treated over I think 20 kids with ADA SCID, and they've seen an immune reconstitution. So SCID is the immunodeficiency. So this is done on a normal basis now. So we would isolate these stem cells from the patient, and then we would genetically modify them ex vivo. So this is an ex vivo gene therapy where we take the cells out of the body and we genetically modify them outside prior to putting them back in. So it's different than injecting the vector into a patient. The patient would then get a conditioning regimen similar to a bone marrow transplant, and then the UBE3A expressing cells would just be injected IV. As I mentioned before, these cells, they reside in the bone marrow, but a small percentage circulate in the blood, but they know to home back to the bone marrow. They know where they're supposed to live. So a straight IV injection is all that's needed, and then they'll go back into the bone marrow, engraft, and then they'll start making a new immune system that will now express functional UBE3A throughout the body. So just as a review of the actual concept, we're using an insertional gene therapy strategy, using a lentiviral vector as the gene therapy vehicle. It's a modified UBE3A, because if we just put the normal UBE3A in there, it wouldn't be secreted from the donor cells and it wouldn't be taken up by the recipient cells. And then the treatment strategy would be a stem cell transplant, and we would use the blood system to transfer this UBE3A throughout the body. So now on to a little bit of the data. So in collaboration with Dave Siegel's lab, so we first had to look at the functionality of this modified UBE3A. Because we modified it, we need it to still work like it normally would in the body. So once it gets into the recipient cells, it works properly. So the figure on the left shows expression of the overexpressed UBE3A in the gene-modified cells, and the figure on the right shows the actual functionality. So you can see those bands on the right side of panel B of the slide. You can see that ladder effect of the bands, and that's what UBE3A does to a target protein. It ubiquitinates it, so you see that ladder effect. So this modified version of UBE3A was still able to function on one of its target proteins, the S5A. I'm not going to show the safety data, but the bullet at the bottom just explains that safety data was obtained in an in vitro assay, so in a cell dish assay in the lab, showing that these stem cells, when gene modified with this modified UBE3A lentiviral vector, they were still able to differentiate and proliferate in a normal way. So the expression of this, the overexpression of this gene did not affect its differentiation in any way. And then the next big step in our project was to generate an immunodeficient UBE3A mouse model. So my lab works with the human stem cells. There are mouse stem cells, but when we're developing therapies using human stem cells, it's nice to use as close to the therapy as you want when you're doing your preclinical studies. So there's less hurdles less hurdles and other experiments that the FDA may require you to do, because a mouse stem cell may take that protein that you put in it and do something different to it than a human stem cell might. So we want to use as close to the therapy as possible. So we needed to generate an immunodeficient UBE3A deleted mouse model, so we could then transplant human stem cells into this mouse. And since the mouse is immunodeficient, it doesn't reject those cells. So you've probably heard of graft versus host disease. If you get a transplant from some other donor, you don't have the same cell surface proteins, so sometimes your body will reject that. But in this case, these mice do not have an immune system, so they don't reject the human stem cells. So this work was done, and the next few slides were done in collaboration with Jill Silverman's lab. So the way that this works, I'll just go through this real quick. We can take cord blood or bone marrow stem cells. We separate them, and then we're able to transplant them into these mice. And then about 12 weeks later, these mice have a complete and functional human immune system. So now we're able to genetically modify these human stem cells with this lentiviral vector that expresses this UBE3A, transplant it into the immunodeficient UBE3A deficient mouse model, and then look for phenotypic recovery. This is a slide just showing kind of the hardships that we've had over the past year in developing this mouse model. Actually, some of the immunodeficient genes that we use in these mouse models, they showed similar phenotypes to Angelman syndrome. And so the middle mouse, this RAG2 mouse, we get great engraftment in this mouse, but it actually shows AS phenotypes by itself without even having a UBE3A deletion in it. And so since we need to look at those phenotypes, we can't have a mouse that shows Angelman phenotypes without having the Angelman genotype. And so we finally came across the IL-2 knockout mouse. So that's the bottom mouse in the graph with the green arrow. Great engraftment in this immunodeficient mouse model, but it shows no AS phenotypes. So this was crossed with the UBE3A deleted mouse model, and we were able to show a lot of the phenotypes that are seen in the UBE3A mice. So on the left, there's a spike in the delta wave, so this just shows susceptibility to seizures. This is the beam walking. No, sorry, this is the digigate. I'm trying to get this to play. All right, well, the mouse on the left is a wild type mouse. It walks very fast. Dr. Weber showed a video of this, so you guys would know what this would look like already. But the mouse on the left walks with a fast, normal stride, normal gait, and then the mouse on the right is a lot slower, has the wider gait, and less stride frequency. And so after making this immunodeficient UBE3A mouse model, we then had to show that it still had those same phenotypes. So when we transplanted them with the cells, we can look for the correction. And this graph shows, again, motor ability of the immunodeficient UBE3A mice. This is an open field assay showing horizontal activity, vertical activity, total activity, and you can see that the dotted line on the bottom are the UBE3A deficient mice. So it just shows that these mice do have these deficits compared to wild type mice. And then this is the slide that we're excited to show everyone. We've actually seen improvement in the motor and behavioral skills in one mouse that was transplanted with the genetically modified stem cells. And it is one mouse, but we are very excited about it because it's not like we tested 50 mice and only one showed this. This was actually the first mouse that got the genetically modified cells, and we see this great improvement in the motor and behavioral skills. So the focus is on the blue bars and the blue line. That's our one mouse that we have so far. The left panel is beam walking, and if you compare the blue bar, that's our transplanted mouse, to the black bar, that's the UBE3A deficient non-transplanted mice, there's great improvement in beam walking, as is rotarod in the middle. You guys have learned a lot about rotarod today, so I won't explain it, but the blue line is our transplanted mouse. And the nice thing about this graph is that the mouse started as low as the dotted line on day one. So the dotted line is a non-transplanted UBE3A deficient mice. So it started low, but on the second day, it actually jumped up, and that shows not only motor and behavioral, but it also shows learning. There's some learning in rotarod, and then on day 3, it stayed high as well. And then in the digigate, if you compare the blue bar to the black bar, the blue one is a lot better, which is our transplanted mouse. And so as far as, so all of these studies are blinded, and what I mean by that is that when I transplant the mice with the gene-modified cells, I don't know what their genotype is. So when we get a litter of mice, there's a mix of wild type, and there's a mix of UBE3A deficient mice. And I don't know that when I'm transplanting them. We don't want to introduce any bias. And then when I transfer these mice, once they're old enough, to Jill's lab to do the behavioral studies, she doesn't know what the engraftment is, and I don't even know what the engraftment is. So they're doing these behavioral studies without knowing what cells, what mice have, which is great, not to introduce any bias. So once all the behavioral assays were done, I went over and we bled the mouse to look for the engraftment, and the engraftment was great. And I was surprised because I work with a lot of these other mice for the lysosomal storage diseases, and I've seen the UBE3A deficient mice, and this mouse, you know, it reacted to me trying to grab it by its tail, and it was running around, and it looked like a normal mice. So we double checked the genotype, and it was a UBE3A deleted mouse. Like, we were a little worried if the genotyping went wrong, but everything looked great. And the engraftment was, there was about 35% human immune cells in this mouse, and about 52% were gene modified. So there was around 20% of the mouse's white blood cells circulating throughout its body that had this overexpressed form, modified form of UBE3A. So it was definitely good to see that we had such a large number of these modified cells in there that may be contributing to this motor and behavioral increase. And so it is only one mouse. This was the first cohort that we checked. As I said, we don't know the genotype when we first transplant these. And so this was the first group of mice, but we currently have around 25 other mice that are transplanted, 12 of which will enter the behavioral studies at the end of this month, and then a couple weeks after, there's another 12 or 13, and as new mice are born, we transplant them with the gene-modified cells, and then Jill's lab does the behavioral. So hopefully there are more mice to add to the blue section very soon. We're very excited about this. As Allison mentioned, we started in January 2017, had to do the vector development, the generation of the humanized mice, and we're right in the efficacy experiment, so where the first asterisk is. And once we get more than one mouse, say 2, 3, 4 mice that show this phenotype, then we'll start overlapping the safety and toxicity experiments, which take 6 to 8 months respectively. And so there's kind of an overlap between the efficacy and safety and toxicity experiments. And then after that, we'll set up a pre-IND meeting with the FDA to go over the data. Once we submit the package, they take about 60 days to respond for a pre-IND, and that will tell us if we need to do any more experiments, any changes in the clinical protocol. So you submit a clinical protocol on how you would treat patients with this. And so I should mention that we're currently evaluating the transplanted mice with mouse isoform 3. So there's different isoforms of UBE3A, and just based off of recommendations from the FIRE team, mouse isoform 3 was the best one to use, and that's homologous to human isoform 1. So we would do the safety and toxicity studies with the human isoform because that's what the FDA would want to see. But as far as the efficacy experiments, they're done in the mouse to look at the phenotypic increases. And so we have to use the mouse isoform for those studies. So in the end, I'd like to thank everyone in my lab, Julie, Haley, and Janine, everyone in Dr. Silverman's lab, Anna, Nicole, and Heather, who do a great job with all the behavioral and the breeding of the mice, and then Dr. Siegel and Henriette for helping with the expression and the functional assays of these vectors, and of course FAST for funding this project, and everyone at UC Davis. So with that, I'll take any questions.
Any questions?
Thank you very much for that terrific work. Just a question about the patient conditioning that you mentioned in comparison to a bone marrow transplant. Were you referring to that just because they were human cells in a mouse, or even in a human patient receiving their own cells back, would they need conditioning so the new cells aren't crowded out by the bone marrow? Would it have to be a much less aggressive conditioning than a human receiving a bone marrow transplant?
Yeah, so that's a great point, and I didn't mention it, but this would be an autologous transplant, so we would use the patient's own cells. There's far less risk in that. And then I've talked to the transplant physicians about this, and with a deletion, you don't have UBE3A in the body. So the body may look at our modified cells as foreign because they're expressing this protein that they've never seen. So as far as a full conditioning, that's what they've suggested. For some of our other clinical trials that we're going to start, like for Tay-Sachs, some of the clinicians who've done cord blood transplants in Tay-Sachs patients, they've recommended a full conditioning as well because the body would recognize that protein as foreign because it is throughout the entire body. So when you do a full conditioning, you're sort of rebooting the immune system more than if you do a slight conditioning.
Is there a problem doing that in children, or is that okay?
I think there's been protocols that have been worked out. Like I mentioned, Don Cohn's lab, he's done a number of infants with ADA skid, and he's an advisor on our HIV clinical trial right now, and so I'm sure them and people at UCSF we're collaborators with where they have protocols. At UC Davis, we don't have an infant transplant group, so we collaborate with UCSF, and they do those.
And also just to mention the other diseases like metachromic leukodystrophy and adenoleukodystrophy, where they did the same thing for neurodegenerative disorders. Those were done ideally as early as possible at diagnosis when they're very, very young. No, those aren't SCID. No, those are not SCID. Those are a neurodegenerative disorder. Yeah. Any other questions? I have a quick question. I'm sorry, I'm just going to ask this real quick. Many of us, because of the decade that our children were born in, saved cord blood. And so we have cord blood for many of our children with Angelman syndrome that is stored, and we pay a tremendous amount of money every month to maintain that cord blood and to cryopreservation. Is there any benefit that, because a lot of people talk about if they should save the cord blood of their children, and if they have it, what they should do with it, and if it could benefit them in treatments in any way. Is there any feeling that you have there in terms of the benefit of using their own bone marrow versus using their own cord blood?
Depending on the volume of the cord blood, you would typically get more of the stem cells out of the body rather than from the cord blood. The cord blood cells are definitely a great source of the stem cells because they're more of a premature one than one that's already in the bone marrow. So it would be a good source, but I don't think there's enough cells in there to use as the only source.
And could you benefit from any of them in your research currently?
If the FDA requires us to do some bridging studies with actually looking at Angelman patient stem cells, then yes. So we've been asked for our Tay-Sachs to actually use peripheral blood stem cells from a Tay-Sachs patient just to make sure that the genetic modification works the same, the expression of the therapeutic protein works the same, because with diseases that we don't know everything about, there could be something that a patient's cells may do differently than someone else's.
Okay, I think there was one question over there.
How is the UBE3A, the overexpressed UBE3A, crossing the blood-brain barrier? And why not just synthesize it outside the body and inject the stuff into the bloodstream? Sorry for a naive question.
So does it cross the blood-brain barrier? We do not know that. We know that in our transplanted immunodeficient mice, there are human cells that are in the brains of the mice. But we don't know if those cells are the only source of that functional enzyme that's being secreted, or if protein is getting in and out of the blood-brain barrier. We don't know the answer to that question. And then this potentially could be a one-time treatment. So this mouse that we have, this one mouse, it got one treatment and that's it. And now it has stem cells that overexpress UBE3A. So if you synthesized it outside and injected it, that might work for a little bit of time, but depending on the half-life of that protein, eventually you're going to need another injection, and that might not be bad. But with our approach, it's different. It's a one-time treatment, potentially.
Great, thank you very much.